My name is Dipti, and I'm from Arizona State University. I'm a rising senior majoring in biology. And this summer, I had a great opportunity to work under a Laurent lab at University of California, San Diego. The title for my project is Optimizing a Spatial MicroRNA Assay in Placenta with Antibody Code Detection. So the Laurent Lab specializes in human pluripotent stem cell research and genomics. It works to improve the maternal and infant health. Respective to HuBMAP aim, they are currently researching the function of extracellular vesicles, which are usually collected by blood or urine samples of pregnant women. And this can be used as potential biomarkers to detect pregnancy disorders such as preeclampsia. So before starting the presentation about my project, I would like to give a brief intro about microRNAs. So microRNAs are a non-coding RNA molecule. These are usually small, comprises of 20 to 22 base pairs. These control the gene expression, and it blocks our coding RNA named mRNA. And because of this, it downregulates and suppresses the protein synthesis in the body. This image shows a little bit about the process of it. And these microRNAs regulate maternal physiology and plays a very significant role in pregnancy. Maternal physiology such as implantation or placental development, immune regulation, and fetal growth. The organ placenta secretes these EVs into the bloodstreams, which contains microRNAs, and these can be the potential biomarkers for pregnancy disorders. One of the examples that I found was C19MC. It's a cluster of microRNA which is present on chromosome 19. And this has been established to be pregnancy specific. It can be seen during 4th and 16th week of pregnancy. And this emphasizes the role of microRNA in early pregnancy development and function. So the Laurent lab has performed bulk sequencing before on a sample tissue of placenta, and it has tracked the expression of microRNAs throughout the gestation period. These are some examples of the microRNAs. You can see there are different level of expression over here during different gestation days. The lab performed the technique in situ hybridization, and they were successful with it. As you can see, there are some brown spots over here. These are the microRNA 371A5P. However, this method was unable to determine the precise cell type where these microRNA was located. And this lack of knowledge in cell type identification motivated this current project. So before moving on to the aim, these are the cell types of the placenta. You can see this outer layer, the STB. These are multinucleated, and this helps in exchange for the gases. And the second layer would be the cytotrophoblast, which is CTB. And this helps in development of the placenta. And the next one would be this EVT. These are invasive cells, and these can be found in the bloodstreams, and it ensures the correction of our blood flow in the placenta. So this project has two aims. The first aim was to optimize and validate a spatial microRNA assay, which helps us to visualize and locate the microRNAs within a tissue section of placenta. And our aim too was to combine it with antibody staining to mark specific placental cell type to better understand where the microRNA is exactly located in which cell type. So our aim one was microRNA scope assay. The first step is sample preparation. The second is pre-treatment. The third is probe hybridization. And the fourth one is signal development and amplification. I'll go in detail for each one of these steps. So for sample preparation, we take our FFPE block, and this is a formalin fixed paraffin embedded block. We place this in a microtome, and this slices the block, and each are 5 microns, and these are transferred into a water bath and then embedded into a slide. And then we take the slide to deparaffinize with xylene, followed by ethanol washes. And for the pre-treatment, we use hydrogen peroxide, which blocks the endogenous peroxidase within the tissue. And then the second one is we wash it into the target retrieval. This helps to open up the crosslinks in the tissue, followed by protease. This helps to digest and open up the protein present in the tissue of placenta. After this, we move on to probe hybridization, wherein we use specific probes and add few drops to the tissue. And then we incubate it in the fish oven, followed by the SSC 20X incubation. This is done overnight. And then the next day is followed by our signal development and amplification. This step is repetitive. We use amplifications 6 times, one by one, and then we incubate it in the fish oven, and then we wash it in our wash buffer. After doing this for 6 times, it is followed by red detection, and then DAPI stains. And this step is important because it amplifies the signals of the probes, and the probes are has specific signals, and it is very sensitive. And this step enhances to amplify the number of signals, since these are very low in copies. And we got our results. We first used our control HeLa cells. So the fast red stain, as you can see, they're red in color. They stained the RNU6 RNA gene. This gene is found in abundance in human body. You can see the DAPI stain. It stained our nucleus. And this is an overlay image of our positive control HeLa cell. We also did it on negative control HeLa cell, and the fast red this time was attached to the scramble, which is basically a jumbled sequence. And then we had our DAPI stain, which is blue in color, and we then got our overlay image of our negative control HeLa cell. This was followed by another control sample, but we used our placental tissue as our sample. Same thing again. We stained it with fast red, DAPI stain, and we got our overlay image. This was also done for our negative control placental tissue. We stained our scramble, which you can see there's no red signals over here since it's a jumbled sequence. And we got our nucleus stained in blue in color, and we got our overlay image over here. So moving on to the next aim, this was supposed to combine antibody stains with the assay. These are the cell type we are focusing on, and these are the respective antibody and 2 of the stains. These are going to fluoresce all these cell types over here. So before starting with the experiment, we had to choose a specific antibody conjugated to a specific fluorophore. And for selecting these fluorophore, we had to understand the spectrums of these fluorophores. So the first step was to consider the laser availability. This has to match with our imaging system. So we chose 405, 488, 561, and 663 lasers. And this was followed by considering the emission and excitation spectra. This is a bit complicated image, so I have this image to break it down. You can see the blue line over here, which is our excitation line. This is when the molecule absorbs the light and it moves from ground state to its excitation peak. And then after a brief period of time, it releases its energy and then hits back to the ground state and emits light. And this emission, this spectrum is called as emission spectrum, and we visualize these spectrum. And then the other thing is our Stokes shift, which is a distance between excitation and emission peak. This helps to enhance our visualization and reduce our background noises. So after going through these steps, we found our fluorophores, and we decided our laser excitation for each one of them. However, one more important thing was to avoid emission spectral overlap. And from the previous slide, you saw that the Pan-CK and HeLa-G cell antibody excites in 405 laser. So I have an image over here which just shows the emission spectrum of HeLa-G and Pan-CK. And as you can see, there are some areas which do not have overlap. This tells us that we would be able to see our fluorescence under the microscope. So after deciding which fluorophores we would use, we continued our experiment. We did a microscope assay, and then it was followed by PBST washes. Then it was blocked in co-detection antibody diluent and was incubated. And again, later on, it was incubated in antibody solution, which comprised of co-detection antibody diluent and antibodies conjugated with fluorophores. Then again, this was followed by more PBST washes, and the final step was the placement of cover slip with vector mount, and then it went forward with the visualizing process under confocal microscopy using Zeiss LSM 780. And these are our results. We got some pretty images. This is area 1 for microRNA 126.3b. It had an expression in later gestational period. And I have a split image over here for you guys to see all the stains with specific cell type. And this is our microRNA 126.3b. And as you can see, this matches with our CD31 stain, which is our endothelial cell. This gives us an idea that our microRNA 126.3b is located in endothelial cell. This is an area 2 for the same microRNA. And the split image for this. And you can still see the microRNAs are located in endothelial cell over here. So in conclusion, we found out that microRNA 126.3b is located in endothelial cells of a placental tissue. Here is an overlay of just microRNA and endothelial cell. And we come to our conclusion that for AIM-1, we were able to optimize a spatial microRNA assay, and we visualized the microRNAs within a placental tissue. And our second aim was to combine these antibodies to mark specific placental cell types, and we were able to mark them, which were our endothelial cell in the case of microRNA 126.3b. And our future direction is to limit crosstalk between channels. These may have to investigate spectral unmixing. And the lab would investigate more gestational ages and more microRNAs to get more idea about the microRNAs they have found till now. And using image analysis software such as HuBMAP would help us to quantify the number of microRNA abundance across the tissue. And I would love to give acknowledgement to Dr. Laurent for selecting me and giving me this opportunity to be an intern at our lab, and Dr. Scott for being an amazing mentor and guiding me all over the process and being patient with me. And Annalise was such an inspiring lab mate who taught me how interesting a microscope can be. And I would like to thank NIH for funding me for this project and all the board members of HuBMAP team for being such great mentors and helping us all the way through this internship, and UC San Diego and Arizona State University for supporting me. I would also like to thank my family and friends to support me through this journey. That's it. Thank you. Do you guys have any questions for me?